Hi, my name is Leah Leibs, and today I'll be discussing the research that my lab partner Nathan Schneider and I conducted under Dr. Melissa Kosinski-Collins in the Department of Biology at Brandeis University in Waltham, Massachusetts. The research is entitled Negative Geotaxis Assay Finds Impaired Locomotor Activity in PolyQ128 Expressed Drosophila melanogaster. It is well known that the gradual onset and progressive effect of Huntington's disease is caused by aggregation of HTT residues in neurons due to a mutation in the HTT gene that causes 36 or more CAG repeats to appear within that gene. This mutation often leads to locomotor impairment and memory loss. My research partner and I decided to look at the correlation between age and locomotor impairment by incorporating 128 CAG repeats within neurons into 1 and 2-week-old Drosophila melanogaster using a negative geotaxis assay. As you can see in figure 1, we had 4 distinct vials, 1 and 2-week-old males and 1 and 2-week-old females that we were then able to use and to test with a negative geotaxis assay. In order to test our hypothesis of and the proven fact that there's a correlation between age and number of CAG repeats, we had to incorporate the repeats to begin with into the flies because they're not inherently found within the flies, and we used the UAS-Gal4 driver-promoter coupling system in order to do so. And in terms of setting up our experiment, we separated out all the different phenotypes of flies and had each type of fly within a graduated cylinder be able to run around, climb up the sides of the cylinder for up to 15 seconds, and then we assessed how many flies were able to either meet or surpass the 30 milliliter benchmark point. As you can see in figure 3, the largest percentage of flies that were able to meet or surpass the 30 milliliter benchmark were the 11 male Q128 1-week-old flies that did not express the PolyQ128 repeats. The smallest number of flies that were able to pass or meet the 30 milliliter benchmark were found by the 22 female Q128 2-week-old flies. This research seems to suggest and imply that it is true, in fact, that the neurodegenerative effect of excessive CAG repeats would cause locomotor impairment because the expressed repeats inhibited the female fly's ability to move as well as the male flies that did not have the expressed repeats within them. A further step within this realm of study could look at 64 repeats, exactly half of the amount that we looked at, and see if there really is a direct correlation between 1 and 2-week-old flies and 64 repeats to see if a smaller number of repeats would lead to a slower onset of this disease. I would also like to acknowledge Dr. Mermanesh, Dr. Piasta, Abraham Chelov, and among others, as well as the Department of Biology at Brandeis University for funding this experiment and for their continued support and help throughout this experiment. Thank you for listening.